now is lecture 5, the nervous system. Nerves tingle and tell us of nerves tingle and tell us of what we are, but not what are we but nerves. Not but. Whoa, Monday morning. Nerves tingle and tell us of what we are, not but what are we but nerves. I think I need a comma in there somewhere after the not. All right, the nervous system. Here we are. Our pal Galen there. So if you'll remember, I had said, I talked about how Aristotle in the 4th century BC had thought that nerves originated in the heart. And this was, of course, a mistake. The word brain itself, which is the central element of the nervous system, the word comes from a Proto-Indo-European origin, meaning Moregno. I don't know how you say that. I don't really speak Indo-European. But it meant skull or brain. And then eventually that word translated into this Proto-Germanic, an ancient form of German, Bragnan, which is where we get brain. The point of that is that the brain has been something that, even though it is this intimate organ in the body, it's something that humans have placed importance upon going back very, very early and having a discrete word for that organ in the center of our heads. It wasn't until Galen in the 2nd century AD began to bring a little more order to our understanding of what the brain is. Quote, who know, he disparaged those who, quote, know nothing of what is to be seen in dissection. Again, the plug for his book here. In my book on the teachings of Hippocrates and Plato, that the source of the nerves of all sensation and voluntary motion is indeed the brain, the encephalon in his parlance, and that the source of the arteries and the innate heat is the heart. So you can see by this quote that that Galen was, first of all, a pretty tough cookie, and also that as he disparaged Aristotle, Plato, the ancient Greeks, he also was still kind of beholden to them. He believed in the 4 elements, the earth, air, fire, and water notion of that ontology that was first put forth by Aristotle. And he talked about the innate heat of the body as being from the heart. But there is something interesting suggested in that we do have thermal regulation by the vasoconstriction or dilation of the peripheral arteries that is part of the cardiovascular system. Today, however, it is the central nervous system. So the word nerve or neuron actually comes from the Greek, and it means sinew or tendon, hinting at the misconception by Aristotle. Moses Maimonides, I've talked about him a little bit in the past. He was the 12th century Hebrew scholar that sort of picked up on the theme laid down initially by Galen. Quote, one who is not knowledgeable in anatomy may mistake ligaments, tendons, and cords for nerves, sort of restating what had been said by Galen. Finally, Benedetti said, by means of nerves, the pathways of the senses are distributed like the roots and fibers of a tree, just 5 years after Columbus landed in the New World. So, and here we begin to see that there's this understanding that nerves themselves are the pathways for sensory information. If you interrupt those nerves, you lose the functional input to the central system. All right. Leonardo da Vinci in the 15th century, also in the 15th century, says that the medulla is the source of the nerves which give voluntary motion to its members. So he saw the medulla as being the point of radiation for all of, indeed, the cranial nerves, and as well as where the spinal cord descends and gives rise to all the spinal nerves. Indicating also that it's not just sensory nerves. So Benedetti is talking about sensation, afferent nerves, bringing information into the brain. Leonardo is looking the other way, seeing that these nerves are efferent, or motor nerves, signals descending from the brain and giving control to the various effectors, specifically the body, the muscles in the body. All right. So this is the basic understanding that we have now, that there is a sensory function, an integrative function, and then a control function. Benedetti and Da Vinci looking at either sides of this coin. Benedetti looking at the sensory part of this cycle, the collection of information that then goes up to the central nervous system, the integrative part of the nervous system that processes all of the information. And then after integrating that input from the sensory receptors, then sends out some sort of control signal via the efferent nerves. All right, so before we get into it a little bit, let's look at an overview of the nervous system. So the nervous system itself can be broken into three general categories. There's, of course, the central nervous system, the two parts of which are the brain and the spinal cord. So the central nervous system is where information gets integrated. It's the processing center for information. Nervous system. Then there's the peripheral nervous system, which itself is broken up into two segments. There's the somatic nervous system. This is the part of your peripheral nervous system that is under voluntary control, voluntary awareness, or voluntary control, depending on whether it's afferent or efferent. And then there's the autonomic nervous system, the part of your peripheral nervous system that is not under voluntary control. You do not choose to make those nerves do what they're doing. That is broken into the sympathetic and parasympathetic nervous systems, the fight or flight, and the sympathetic nervous system, and then the rest and digest, parasympathetic nervous system. And then lastly, the third branch of the nervous system that is marginally autonomous. So this nominally autonomous nervous system is called the enteric nervous system. And the enteric nervous system essentially inhabits the back wall of the abdominal cavity, the abdominothoracic cavity. And its function is to regulate the digestive activities of the viscera in the ventral body cavities. It does have crosstalk with the other parts of the nervous system, but it's minimal, and it's able to function essentially on its own. Your enteric nervous system can function reasonably independently of the other parts of your nervous system, which is why, for example, a person who becomes paraplegic is able to still have digestive activities happen in their body, for example. All right, so that's the basic overview. Now let's look at the kinds of neurons, the kinds of nerve cells that inhabit this hierarchy. So all of these different boxes on the top are just made of neurons, various neurons, and we can categorize them in a number of ways. There are actually three dichotomous distinctions that we can have, three dichotomous distinctions. The first of them is is the neuron a general or a special nerve? Is it a general or a special nerve? And a general nerve is going to be, for example, a touch receptor in the hand would be a general neuron. Or a special neuron may be a photoreceptor in the back of the eye related to the special nerve. Then there is the somatic or visceral distinction. Somatic meaning body, the soma means body, and visceral meaning your organs. And in general, that distinction is kind of roughly the same as saying voluntary versus involuntary, somatic being voluntary, visceral being involuntary, or somatic meaning the outward-facing sensations versus visceral being the inward sensations. And then afferent or efferent. I've been using those words the last few minutes here. But to define them for you, afferent is a nerve that takes information and directs it, collects some sort of information, and takes that and directs it towards the central nervous system. And an efferent nerve is one that takes its stimulus from the central nervous system and sends it out to various effectors. So the prefix there, afferent versus efferent, is the same in terms of meaning as affect versus effect. Yes? Yeah. Somatic would be like a somatic efferent nerve, for example, would be a motor neuron that causes a muscle to contract. A visceral efferent may be a nerve that goes into your suprarenal gland and causes it to make cortisone or something like that, for example. All right, does that make sense? Yeah. The somatic tends to be musculoskeletal or skin related, like more exterior facing, and then visceral tends to be more interior or visceral orientation. So like to be affected by something, something that's going on causes a change in you. It comes into you, whereas you cause an effect on something. You are going to effect a change in your surroundings. That's something you pointing outward. So an afferent neuron is sensory information that's coming up into your nervous system, and efferent is a motor neuron that is sending a signal for action out of the central nervous system. Does that make sense? All right. So because there are 3 dichotomies there, you can get 8 combinations out of them. You can have general somatic efferents, for example. That's the motor neuron that I talked about. Or you can have special visceral efferents. That would be like a neuron that would make you cry, for example. There's all these different combinations. There's in fact 8 of them, maybe 7 general special efferent and special somatic efferent. There's a gray area between. But all right. Is there any questions? Other questions on that? All right. So first, functionally, let's look at these classes of neurons. I've already sort of talked about this a little bit, but there are these sensory neurons. The first part of the loop that I showed on the first real slide here was the sensory input that's going up towards central processing. These are afferent neurons that are part of the peripheral nervous system. And they can be categorized into 3 general types. We can have interoceptors. Interoceptors are going to be ones that are going to monitor some sort of internal state. For example, your digestive system, the respiratory system, cardiovascular, urinary, reproductive system, etc. So you ate a bit of breakfast and the food ends up in the stomach. Interoceptors are going to be ones that are triggered by the fullness of your stomach, for example, and send a signal up. And then also the internal senses, taste, deep pressure, and pain. All of the more internal sensations that you may have. Those include taste, deep pressure, and pain. Then that is in contrast to the extero or external ceptors, exteroceptors. These are your external senses such as touch, temperature, pressure. So light touch, light pressure, temperature, and any of the distant senses like sight, smell, hearing. Right? So intero versus exteroceptors, and that formalism sort of lays on top of the categorization that I just showed you in the last slide. Finally, there are sensory receptors that we call proprioceptors. And the proprioceptors are going to be ones that are going to monitor the position of your body. So they're going to relate to the skeletal muscles and the joints. You'll find them, for example, a muscle spindle apparatus can be embedded in the connective tissue of a muscle. We talked about, in the muscle chapter, I talked about the paramecium, for example, of a muscle. You'll have a muscle spindle embedded in there, and it's going to tell you whether that muscle has rapidly contracted or not. It's these sensory neurons, these afferent neurons, that get triggered during the reflex test, for example. So that's the first part of the loop, the afferent neurons. The second part is composed of interneurons. So the interneurons are the ones that are the principal component of the central nervous system. And they're going to be associated with all of the higher functions of the brain and central nervous system. So integrating information, perhaps in some sort of homeostatic system. We in the very beginning of the class, we talked about the temperature control, the homeostatic feedback. The integration center would be made up of interneurons. Inter simply referring to the fact that they are in between sensory and motor neurons. Memory, learning, all of the great things that make you you are due in some way to interneurons. Now, interneurons make up the vast majority of the neural tissue in your body. Most of the neurons in your body are made up of interneurons, and I am going to talk about them just very little. I'm going to give the tiniest amount of attention to them because they really requires a neuroscience class to get into that. But we'll talk a little bit about it. And then finally, motor neurons. Efferent neurons of the peripheral nervous system. These are simply relaying the information to do something to an effector. An effector is some sort of organ that is going to effect a change. An effector can be a muscle, for example, or an effector can be a gland, such as a salivary gland or a lacrimal gland, or there's a bunch of different glands that have nerves that go into them. Some kind of effector is stimulated by an efferent neuron. So in general, as I already pointed out, there are two categories that are somatic efferent neurons, which end in the skeletal muscles, and then everything else is considered to be a visceral effector. And visceral effectors are ones that are under the control of the autonomic nervous system, so the sympathetic or the parasympathetic nervous system. And I don't know how much of the nervous system I'm going to get to cover, but I'm definitely going slower this year. But the general structure of the autonomic nervous system has what are called preganglionic and postganglionic fibers. So that implies that there are these things called ganglia in the autonomic nervous system, which are basically just balls of cell bodies where there's a synapse distributed throughout the body in different places along the sympathetic chain for the sympathetic nervous system, and then what's called paramurally next to the organ that they innervate in terms of the parasympathetic. But hopefully we'll get to see that in a few lectures from now. All right, questions about that? So I'm just giving you a very broad overview of the functional and structural categories here of different neurons in the nervous system. So let's look at the nervous system itself, the central nervous system itself in a little more detail. We have the brain and the spinal cord, I said, were the two parts of the central nervous system. Let's consider first just the brain. So any kind of sensory receptor that's going to be bringing information directly into the brain is going to be bringing that information into the brain along what is called a cranial nerve. A cranial nerve. There are 12 sets of cranial nerve. There are 12 pairs, left and right. There are 12 pairs of cranial nerves. The cranial nerves are called cranial nerves because they enter the brain directly through some sort of hole or foramen, some orifice in the cranium. This is why we call them the cranial nerves. And then any of the motor output that comes out of the brain directly is also coming along one of those 12 cranial nerves. Cranial nerves themselves, I used to go through them in way more detail, but I'll show you at least one slide about them. These cranial nerves can be mixed nerves, meaning of those 8 categories of fibers, I said there's like G, S, E, J, G, S, A, etc. The general, somatic, the special, the visceral, all that, afferent, efferent. Of those 8 categories, any cranial nerve they're pretty much all, I believe, mixed nerves. Is that right? No, not all of them. The optic isn't, nor is the olfactory. But most of them are mixed nerves that are going to have multiple fiber types, some afferent and some efferent in them. Okay. And then there's the spinal cord. If information gets into the central nervous system via the spinal cord, it's coming in via what are called spinal nerves. So both cranial nerves and spinal nerves are considered to be part of the peripheral nervous system. All right. Spinal nerves are the ones that most people are familiar with. So, for example, the vast majority, although not all, but the vast majority of muscular contractions, for example, are propagated via spinal nerves. Certain muscles of the face, for example, are controlled via the cranial nerves. But most muscular contraction is by spinal efferent nerves. And then all of this motor output ends on some kind of effector. And I had talked about this a moment ago. Effectors break down into muscles and then glandular tissue, glandular epithelium, and also adipose tissue actually is considered to be an effector under the control of nervous tissue. All right. Any questions on that before I move forward? Broad strokes here. We'll fill in some of the details, sketch it out a little more, but we got to give you some more general information first. So, let's start going our way through the same loop that I've been talking about since the beginning: sensory up to integration down to motor. We're going to go through sensory first in more detail. So any kind of sensory receptor is going to transmit 4 kinds of information. Every one is going to transmit 4 kinds of information. The first is modality. Modality. And what do I mean by modality? Well, for example, the five senses, taste, touch, hearing, smelling, and sight. That is the modality of that sensation. In fact, there are more than five modalities. There are sort of sub-modalities. For example, taste itself, we're able to, there are at least five and probably more than five unique taste receptors, all right? And each of them is its own modality. People used to think, there used to be this belief that you only tasted sweet on the tip of your tongue and salt on the side or whatever, but that is actually not how taste works. A taste bud is composed of a bundle of different sensory receptors, and the way that your brain interprets what you just tasted is via what's called the labeled line code. So in this color scheme that I'm showing you here, you see the dark blue sensory neurons represent, they're triggered by alkaloids. So some kind of compound, alkaline compound, that triggers that taste receptor, that chemoreceptor, that chemical sensitive receptor. And then we perceive that taste as bitter because that neuron goes up to a part of the brain. When that part of the brain gets lit up, the brain says, oh, that must be bitter. If you were somehow able to do the miracle of switching around those cables, you could taste something that is bitter, and if you switched it with a cable that goes to sweet, your brain would say, oh, that thing that I just tasted, that horrible bar of soap, tastes sweet to me. It's the labeled line code. So there is a mapping in the brain. There's a mapping in the brain of the different modalities that you sensate. You guys following me here? All right, there's a modality. So what did I say here? Labeled line code. All action potentials are identical. Yeah, so if you eat something bitter, it's not that neuron is firing in a different pattern or something. It's simply that neuron is interpreted as activity in that sensory neuron is interpreted as bitter or salty, etc., or sweet, sour, which is acid, bitter is alkaline, and umami, which is savory. Umami is triggered by glutamate and other acidic amino acids. So, all right, there's that. For example, in touch, there's multiple modalities. There's light touch, there's deep touch, there's vibratory touch. There's all kinds of different touch modalities. So anyways, the first kind of information that a sensory receptor is giving you is the modality. The second is location. Where on the body is it happening? If it's a sensory receptor, for example. So the first thing that this implies is that there is some kind of what's called somatotopic map. And we're going to return to this idea on Wednesday of the somatotopic map. Meaning that there's a little map of the body in the brain, and when a neuron wherever on that map that corresponds to your hand, when that lights up, your brain is able to interpret that as being in the hand and not on the elbow or on your cheek or something. An important concept here is the idea of receptive field. And we're going to come back to this idea when we get to talk about a guy named Wilder Penfield and his homunculus, which will probably be next time. But the idea here is that a neuron, a sensory neuron, we'll just say a tactile neuron, is going to have a certain square area, a certain range that it monitors within this somatotopic map in the brain. So, for example, you can take these calipers here. You see the woman with the calipers on her shoulder, and the sensation on your shoulder is actually not highly resolved. It's not highly spatially resolved. So you can have the calipers be quite far away from one another, poke them on the back, and that person may only feel that they're getting poked by one needle rather than two, because those needles are stimulating the same sensory neuron. All right. Whereas if you were to take those calipers and at the same width poke the finger or the lips or anything like that, you would be able to have the distinction that it is a single pinprick. And you can map out the density of the neurons in a part of the body using these kind of calipers. I do that in the lab in 265. But all right, so this is to say that the size of the receptive field is inversely proportional to your ability to locate that stimulus. All right, so if you have a large receptive field, that's a relatively low spatially resolved neuron. All right, and then you have a harder time saying exactly where that is. You need, and it should make sense because you need fine resolution on places like your fingers, your lips, etc. But on your back, there's a bug. Just get it, whatever. Okay. All right. The third type of information is the intensity. The intensity of whatever stimulus is coming. And this intensity can be transmitted in one of a number of different ways. So the first is which fiber is sending the signal. The brain can interpret a signal from one neuron versus another as being more intense. All right. So in this top example here, the neuron in the middle is interpreted by the brain as being a more intense source of stimulation than perhaps either of the other two. All right. Anytime that one in the middle fires, it's going to have the same intensity. All right. Whereas the other two are, or perhaps it's lower. Perhaps the sensory receptor in the middle has a lower threshold for firing than either of the other two. And if you were to increase the intensity of the stimulus to the point where you fire the other two, then the brain would say, oh, these other ones, these higher intensity neurons are firing now. This must be a more intense stimulus. So similar to the labeled line code. All right. Another way that this kind of information can be transmitted is by how many fibers are sending this information. Is there just one that is sending you light touch, or many sending you light touch? That's going to give you some kind of information about intensity. And then lastly is the rapidity. So when we talked about the cell physiology chapter, I talked about the refractory period. And are we pushing the refractory period of one into the stimulation period of the subsequent action potential? What's the timing of that? What's the timing of that neuron? Okay, so that's intensity. And then the last bit of information is duration. How long is that neuron sending a signal up to the central nervous system? This gives us the ability to introduce the concept of adaptation. So adaptation is simply the reduction in sensitivity to a constant stimulus. There are two types of neurons in this regard. There are tonic receptors. These are ones that are slow adapting. They are always active, and they show very little peripheral adaptation. So, for example, we have proprioceptors. They give you fairly constant information about the position of your body, muscle tension, joint motion, etc. So when I walk over here and I pick up a book, I'm holding this book as I'm talking to you, and if I were to continue talking to you, the whole time my brain would be aware of the tension in my muscles here that I'm holding this book. I don't forget that I'm holding the book. These are tonic receptors. They are extremely slow adapting. On the other hand, there are phasic receptors. These are rapidly adapting. I run my hand through my hair. And I can feel my hand running through my hair. I've changed the position of my hair, but as soon as my hand is out of my hair and not moving the hair anymore, I am now unaware of the position of all my hair. It has rapidly adapted, even though the position of that hair has been changed and that new position is still in place. It hasn't snapped back to where it was before I ran my hand through it. The information that is being sent to the central nervous system stops. So they're normally inactive, and we use them because to convey a rate of change in a stimulus. So some examples: smell, hair movement, cutaneous pressure. You smell something and then it goes away. You don't continue to smell it long after the odorants have gone away from the sensory receptor. All right. Cutaneous pressure. So you're sitting somewhere. You're not constantly aware of the pressure on your skin, but there is a spectrum here. And if we look at these in this bottom panel, we can see in pink on the very bottom, this is stimulus, some sort of stimulus in a neuron, at the sensory terminal of a neuron. And we look at the receptor potential. We stimulate the neuron, the tonic receptor comes on and equilibrates, but stays essentially on for as long as the stimulus is there. Stays on for as long as the stimulus is there. And then phasic is stimulus comes on, we get a spike that tells us something has changed. But it comes back to the baseline receptor potential. And then you remove the stimulus, and there's a hyperpolarization that is due to the off response of that stimulus. So do I? Yeah, okay. All of this is different than central adaptation. So this adaptation is adaptation that is hardwired, so to speak, in to the sensory neurons that I'm talking about. And it has to do with the types of channels that are stimulating the sensory neuron, what the expression pattern of the various ion channels. But there's also what's called central adaptation. That so as we sit here, let's take a moment of silence here. A lot of sounds. I hear that rattling, don't you? I hear the fan on my computer here. We're completely surrounded by some sort of hum or rattling or something all the time. And our brain has learned to filter that stuff out. Our brain is particularly good at recognizing patterns and trying to pull patterns out of the noise, patterns out of the chaos. People think that your brain is all about collecting as much information as possible. In fact, quite the opposite. The brain The brain's job is about weeding out as much as it possibly can to find just what's meaningful to it. All right, and that's called central adaptation. Our ability of our brain to filter out meaningless sensory input and just find information that's meaningful. So those are two different things. This is peripheral adaptation. That's central. Okay. So here are the various classifications for these afferent neuronal categories. And I've broken it down. I used to have a slew of like 35 slides that outlines all this, but for the sake of time, I'm just going to kind of go through the outline. It doesn't really drive the material that much better to have all those slides. So first of all, there were the fiber types, and we talked about the fiber types on one of the first slides there. General versus special, somatic versus visceral, afferent versus efferent. And since we're talking about afferent neurons here, only the top four in that red box there, which are then in a little box of green and blue, are the ones that we're talking about. General somatic afferent, general visceral afferent, and special somatic afferent and special visceral afferents. There's the distribution of these fibers. The distribution is a reflection of intero versus extero versus proprioceptors. We can classify them by modality. The general versus the special have different sets of modalities. The general senses are temperature, pain, which is called nociception, N-O-C-I-C-E, pardon me, P-T-O-R. A nociceptor is a pain receptor. Touch, pressure, vibration, and proprioception. And then the special senses are olfaction, vision, taste, equilibrium or balance from the inner ear, and finally hearing. And then there are functional categories. There are functional categories that all of these afferent neurons can break down into. The first functional category are pain receptors, nociceptors. There are going to be fast type A, slow type C, and these sort of intermediate, which I didn't include, type B fibers. But nociception is pain. Thermoreceptors are going to be receptors that are triggered by a change in temperature. Third class would be chemoreceptors. These are afferent receptors that are triggered by a change in pH or a partial pressure in carbon dioxide. For example, there's a lot of different types of chemoreceptors. Taste, smell, those are both going to be their own categories of chemoreception. And then finally, mechanoreceptors. And these are ones that are triggered by physical distortion of some type. Some kind of physical distortion is going to trigger that afferent neuron. And that is, the categories of those are tactile, touch receptors. There's a slew of those. Baroreceptors, which are pressure receptors. For example, the baroreceptors that are in the carotid bulb or the aortic arch that monitor your blood pressure. Proprioceptors, we've talked about, returns information on physical orientation, such as the muscle spindle apparatus, the Golgi tendon organs, or the various receptors in the joint capsule. The tactile receptors, touch receptors, run this spectrum. I have them listed there from free nerve endings all the way up to what are called Ruffini corpuscles. There's a couple others there that I didn't include. But free nerve endings are ones that the neuron just has projections into whatever tissue we're talking about, but there's no associated structure with it. A root hair plexus would be the same thing, except it's now wrapped around the bulb of a root hair, And then these other ones, as we go down this list, have accessory structures around them they're sort of modulating the kind of stimulus that is triggering those tactile receptors. So, for example, a Meissner's corpuscle. These are going to be the soft touch ones that are highly concentrated in the lips, the fingers, the external genitalia, whereas the lamellated or Bassinian corpuscle would be deep pressure that you would find at the hypodermis. That would give information about how much pressure is being pressed on yourself. All right, I think that's a good place to stop. We'll pick up next time. So that was the functional categories of afferent neurons. Next time, we're going to do a little bit about interneurons. We'll go through the structural classification of interneurons. We'll walk through the central nervous system a little bit, and then I'll talk about motor neurons as well, and we'll be done. On Friday, I think I'm going to do this on Friday. What I would like to do on Friday is meet with the groups at like different 10-minute blocks of time during class instead of having class. And what am I going to do? I'm going to put, I don't know, I'll put some kind of way for you to sign up for blocks of time so that you know when each group. I don't know how I'll do it, but there'll be something. I'll look for an email through the Moodle as a way to sign up for a block of time for Friday to meet with me, okay, during that class. Thank you very much. Thank you very much. Thank you very much.